We just did our project on ketone body production and starvation. These are the 6 topics that we're going to be talking about in this video.
Okay, so before we get into it, we're just going to quickly go over how energy is created using carbohydrates, just as a refresher. So if you remember from class, carbohydrates are broken down into glucose, and then that glucose is further broken down into pyruvate, where that pyruvate molecule loses a CO2 group and turns into acetyl-CoA. Acetyl-CoA then enters the citric acid cycle, and then from there goes into the electron transport chain, which is where we get most of our ATP and energy from. This is just an in-depth look at glycolysis. We have this slide here just because this is the main difference between how energy is formed through carbohydrates versus how energy is formed through ketone production.
You might be wondering, what is a ketone body? Ketone bodies are backup sources of energy when glucose is not readily available to be metabolized. There are 3 ketone bodies: acetoacetate, 3-beta-hydroxybutyrate, and acetone. While ketones serve many physiological purposes in the body, they are essential for continuing metabolic pathways during periods of starvation.
Okay, so we've previously discussed how glucose is used from the carbohydrates that we eat to create energy under normal conditions in our body. But under conditions where glucose is not readily available, like starvation, our body has to use different pathways to regulate our energy production without relying on glucose. One pathway is ketone metabolism, which is what I'll discuss over the next few slides. So the liver is in charge of regulating our glucose levels to maintain our energy production, so it's really sensitive to changes in glucose levels. In a condition like starvation, our liver will sense that our glucose levels are extremely low, so it will try to rectify that by entering into a pathway called gluconeogenesis, where the liver will use readily available molecules to create more glucose. Now, there's a lot of steps within this pathway that we really don't have time to cover, but the most important thing to notice is the molecule oxaloacetate is used quite a few times within this process to make new glucose. Oxaloacetate is actually a molecule the liver takes from the citric acid cycle, so in a condition like starvation, where glucose levels are not restored, the liver will exhaust its concentration of oxaloacetate after a while, and the citric acid cycle will not be able to work. This in turn leaves some acetyl-CoA molecules because they can't enter into the citric acid cycle. Luckily, acetyl-CoA can be further metabolized to form ketones to create energy. So along with the concentration of acetyl-CoA molecules from the gluconeogenesis in the body will start to produce more acetyl-CoA molecules by breaking down our free fatty acids into acyl-CoA, and then using beta oxidation techniques to form acetyl-CoA. Okay, so from here, the body kind of uses the high concentration of acetyl-CoA molecules to its advantage, and it essentially does a whole bunch of reactions with acetyl-CoA to make the ketones we've talked about. Okay, so the first reaction that's going to occur is 2 acetyl-CoA molecules are going to bind together and make a molecule called aceto-acetyl-CoA. When this happens, coenzyme A will be discarded as a byproduct. So from the aceto-acetyl-CoA molecule, another acetyl-CoA will come in and react with that molecule to make a molecule called HMG-CoA. Within this reaction, another coenzyme A will be discarded as well. So from HMG-CoA, another acetyl-CoA group is going to come in, react with that molecule, and create the first one of our ketones called acetoacetate. So acetoacetate is polar enough to enter the bloodstream, but from here you can make 2 other ketones. The first ketone that you can make is acetone, which we're all kind of aware of. The way that this happens is there's a non-enzymatic decarboxylation, which means that a CO2 group is lost from our acetoacetate, and acetone is produced. So if you've ever been around someone who's gone into ketoacidosis, you can usually smell a sweetness on their breath. This is because of the acetone that is produced from this reaction, and this is commonly associated with ketoacidosis. The other molecule that is formed that is a lot more useful than acetone is called D-beta-hydroxybutyrate. This is formed when an NADH and an H+ reacts with our acetoacetate, and it forms our DB-hydroxybutyrate. So our acetoacetate and our D-beta-hydroxybutyrate are polar enough that they can enter the bloodstream and travel to our muscle tissues. Okay, so once our ketones enter into our muscle tissue, the reaction for ketogenesis basically occurs but in reverse. So it's basically just undoing what we already did. So the first thing that happens is our D-beta-hydroxybutyrate ketone body is reversed. So if you remember, the acetoacetate molecule reacts with NADH and H+ to form that molecule. So in order to undo that, we just react it with NAD. Once you react D-beta-hydroxybutyrate with NAD, it forms acetoacetate. So now you have your acetoacetate molecule just kind of floating around in your muscle. From here, a kind of weird reaction occurs. Your acetoacetate is going to react with succinyl-CoA. And if you remember, succinyl-CoA comes from the TCA cycle. So once your acetoacetate reacts with succinyl-CoA will turn into succinate, which is the next step in the TCA cycle. And your acetoacetate will turn into aceto-acetyl-CoA. Once again, we're just basically undoing what we did within our ketogenesis steps. Aceto-acetyl-CoA will then be turned into acetyl-CoA. And then acetyl-CoA will enter into the citric acid cycle as it normally would under normal conditions. So the reason why it can enter into the TCA cycle here but not in the liver is because the liver is what actually depletes your oxaloacetate molecule, but in your muscles, gluconeogenesis hasn't occurred. That only occurs in the liver. So your oxaloacetate molecule is here, it's in high concentrations, and the TCA cycle can continue. From here, the molecule can go through the TCA cycle, and then the electron transport chain can continue as it normally would under normal conditions, and energy is produced.
Okay, so I'm going to be highlighting some of the key enzymes and intermediates used and produced throughout this process. So Grace just gave us a super awesome deep dive into ketogenesis, where she already mentioned most of these enzymes and intermediates. But as you heard, it's a very complex process with a lot of key components. So this slide just has it written out for you to make it a little bit easier to understand. So on the left side, we have the enzymes and intermediates involved in ketone synthesis. And then the right side is the enzymes and intermediates that are involved in ketone degradation. So back to the left side, with ketone synthesis, those first 5 steps, those are the intermediates that are made. And then at the bottom, we have the enzymes, which are aceto-acetyl-CoA thiolase. And then that really long word is just abbreviated as HMG-CoA. And we have synthase, lyase, as well as dehydrogenase. And then at the very end, there's a spontaneous decarboxylation. If we move over to the right side for ketone degradation, we have those first 3 steps, which also involve our intermediates. And then below, you can see the enzymes involved there, which are beta-hydroxybutyrate dehydrogenase. Then we have the succinyl-CoA, aceto-acetyl-CoA transferase, and then aceto-acetyl-CoA thiolase.
Alrighty, so I'm going to be talking today about how this pathway is regulated and the various ways that that is done. So there are 3 basic steps or different mechanisms that ketosis and ketogenesis are regulated, and those 3 are 1 through lipolysis or lipolysis in the adipose tissue, 2 through regulation of the oxidation of fatty acids, and 3 through the regulation of acetyl-CoA. So first of all, with lipolysis, ketosis cannot occur without an increase in circulating fatty acids, because fatty acids are the precursor substance for physiologic ketones. So they are the actual thing that ends up oxidized and ends up producing ketones. So if we're going to create these free fatty acids, we need to undergo the process of lipolysis, which is the process of breaking down the triglycerides in our adipose tissue to form free fatty acids. So the diagram on our right here shows how this process goes. It undergoes 3 or 4 different steps, and then it produces fatty acids and 1 molecule of glycerol. Lipolysis is regulated by pancreatic secretions of insulin and glucagon. So it's induced by glucagon and inhibited by insulin. Insulin is a hormone that's released in response to an elevated blood glucose, and it's associated with a fed state. So if we eat food, the energy and the carbohydrates from that food are going to enter our bloodstream, and as a response to that, our body is going to secrete insulin. And so the optimal conditions for lipolysis are going to be a low level of glucose and a low level of insulin, because insulin is going to dephosphorylate and inhibit our HSL here, which is going to make it so that lipolysis doesn't occur and that we don't have a greater level of free-floating fatty acids in our bloodstream. The next method is regulation of oxidation. So basically there are 2 options for free fatty acids. They can either be oxidized or esterified. And the oxidation pathway is kind of guarded and regulated by 2 different enzymes. The first of which is carnitine acyltransferase 1, or CAT1. This basically allows for the free fatty acids to enter the oxidation pathway, but CAT1 itself is regulated by malonyl-CoA, which is formed by acetyl-CoA carboxylase in the fed state. So if we're in the fed state, CAT1 is going to be inhibited by malonyl-CoA, and the free fatty acids aren't going to be allowed to enter the oxidation pathway and end up as acetyl-CoA. And as we have written here, in the fed state, our free fatty acids are going to enter the liver at low levels, and almost all will be esterified and transferred out of the liver as very low-density lipoproteins. Finally, our last regulation pathway is the actual regulation of acetyl-CoA itself. So once we've created acetyl-CoA from that pathway, we have 2 different options that we can go from there. We can either the acetyl-CoA can either enter the TCA cycle and be eventually oxidized to CO2, or we can undergo the ketogenesis pathway and make ketones. So as our serum free fatty acid levels rise, a greater percentage of acetyl-CoA is going to lean towards the left here on our diagram and lean towards the ketogenesis pathway. And so the entry of acetyl-CoA to the TCA cycle is dependent upon oxaloacetate. And oxaloacetate is dependent upon our concentration of carbohydrates, because basically when we have pyruvate, it reacts with pyruvate carboxylase and makes oxaloacetate. So if we have a low level of carbohydrate, eventually we're not going to have enough oxaloacetate for our acetyl-CoA to enter the TCA cycle, and it's going to lean heavily towards the ketogenesis pathway. So quite a lot here for the regulation of ketosis and ketogenesis, but that is a few ways that this pathway is regulated.
Now we're going to talk about some research on our pathway. We found an article called Acetone as a Biomarker for Ketosis Buildup Capability, and in it they were showing acetone as a biomarker for ketosis buildup. This study was conducted on healthy individuals under high-fat and starvation diets. Each individual underwent 3 phases. Phase 1 was a mixed diet with a ratio of fat, carbohydrates, and protein, where 29% of their energy came from fat. Phase 2 was a mixed or high-fat diet. The individuals were separated into 3 different groups, consuming a diet that provides 29%, 79%, or 90% energy from fat. Phase 3, everyone was on a fasting diet without caloric intake. Caloric intake from diets on day 1 to day 6 was equal to each individual's energy expenditure. All of the measurements were taken on day 7, the starvation day. We see here that the breath acetone levels are highly correlated with the amount of blood acetone. We also see that on a high-fat diet with higher amounts of total energy expenditure, individuals have significantly elevated levels of breath acetone and ketone bodies formed than those who are on the lower fat diet with low levels of energy expenditure. We expect to see this because the individuals on a diet with a higher percentage of fat and less carbohydrates will use ketone bodies to get their energy on the starvation day. These are the resources we used to create this video. Thank you for watching.